Good afternoon, everybody, and welcome to today's webinar. My name is Stephan Holl. I'm the Chief Executive at Conscio Group, and I'm very excited to meet so many of you to today's discussion. Our topic for today is expediting biosimilar development, and for me personally, I think it could not be a more timely topic in a period where we see a very high number of biosimilars come to market, and at the same time, we see more and more evolving discussion around the regulation around biosimilar approvals. So this is what we are here to discuss today. And if we briefly look at our agenda, we will be joined by three leading experts in the field. And after a brief introduction about our own group, I will be handing over to Elena, who's a medical doctor and researcher at the Leiden University Medical Center. Elena has conducted wide-ranging research on biosimilar regulation and will share some of her findings and insights on future directions on biosimilar regulation. Afterwards, Martin Baur from Midas Pharma, a collaborator of ours, will talk about common pitfalls on the way from R&D to commercialization of a biotech product. And last but not least, we will be joined by Klemens Weitenthaler of Conscio Group, who will share his views on strategies for a successful bioassay transfer and validation and the associated importance of standardization. We will ask you to put your questions into the Q&A section of the chat, and what we plan to do is to give space for a couple of questions after every talk, and then at the end we will enter into more discussion if and when needed. So without further ado, let me very briefly introduce our Conscio Group. So Conscio is a network of scientific service providers with a focus on the pharmaceutical industry. We've been active since 2021 as a group, but many of the companies in our group have been around for much longer and have been leaders in their field. We've acquired a number of businesses and integrated them in order to become a vertically integrated contract research and development organization, and also a provider of quality control analytical services. We are headquartered in Munich, Germany, where I'm today as well, and have operations also in Austria and the Czech Republic and Slovakia, and are about 500 employees, of which more than 300 are exclusively focused on the pharmaceutical industry. And what we are really trying to do at Conscio is to differentiate a little with not only a particularly high standard on scientific excellence, but also we try to pair this with a relentless focus on customer service. And in that combination, we then partner with our customers in the long run over the life cycle of individual products, from R&D, clinical testing to quality control, and also over many products with the same. Our customers come really from, naturally from Europe, where our base is, but also from the US and from Asia. And we serve everything from rather small clients all the way to quite a few of the top 10 pharma companies in the world. So why biosimilars? We are very excited about our fast-growing biosimilar activities. So there is a legacy of a lot of work with large molecules on the originator space, and for biosimilars in particular, we also conduct this journey from R&D, where we help with method development and optimization, to biosimilarity studies that we can conduct in our own in-house clinical units in the Czech Republic, to the bioanalytical testing of the subjects. And also we are one of the largest quality control service providers in Europe for both small and large molecules. So with this integrated offering, we are having a particularly strong observation on some of the regulatory changes with biosimilars, because we believe that, you know, one aspect of this evolution might be a more common approval of biosimilars solely based on a very high quality analytical testing package paired with a phase 1 study on healthy volunteers. And this is a little bit where we will focus on today. So the market of biosimilars, you know, you're all experts in the field, you're from the industry. I think it's very clear that at the moment we've seen many biosimilars come to market in Europe. And even more importantly, they have been adopted in the patient care with large success. They've been really making their way to patients for better and more economical access to care. So that's very exciting. But now only we will see an additional extremely big wave of products, of blockbuster drugs with peak sales over $1 billion that will lose exclusivity over the next 10 years. So those are, you know, around about 55, a big wave. And at the same time, those developments still are, you know, they're risky, they're very costly. They cost between $100 million and $300 million. They take between 6 and 9 years. So there's still a lot of risk associated with it. And as you see here at the bottom half of the chart, also many projects are abandoned along the way for one or the other reason. So much of those costs, 65% in fact, are in the phase 2 and phase 3 trials. And as the evidence comes along from the early approvals and clinical efficacy studies, there's been increasingly, you know, more observations by industry, by regulators on what the best way for the biosimilar regulatory framework should look like in the future. So that's exactly, you know, the discussion we would like to have with you today from very different viewpoints, and hopefully also in an interactive manner. How can the system evolve to allow for a more cost-effective development of biosimilars and ultimately a faster and more economical patient access? So with that, I'm very excited to hand over to Elena, who will approach this really from the regulatory angle, where she has done extensive research. And over to you, Elena.
Good afternoon to everyone, and thank you very much for having me, Conscio. Thank you also, Stephan, for your nice introduction. And well, I think Stephan already talked a little bit about this, and I think that everyone already understands the need for streamlining biosimilar development. But I do like this image very much because I think it very clearly states the panorama that the healthcare system and the patients themselves will be facing in the next years if biosimilar streamlining is not done. And as we see in the image, in the next 4 years, more than 30 biologics are going to lose their patent protection. And as we can see on the right part of the figure in green, it is estimated that 55%, so more than half of biologics that are going to lose this market exclusivity in this period will be without a competitor. And what this ultimately means is that patients will not be able to have access to innovative and affordable products in the near future. So this really clearly, I think, talks about the urgency and why we're having these type of shared webinars. But of course, it's not all bad news. I'm very optimistic. So I do think that we are in an exciting time where things in the biosimilar landscape are changing very rapidly with regard to the biosimilar regulatory framework. And since the publication of the first guidelines, the European Medicines Agency guideline, et cetera, we have already seen that different stakeholders worldwide believe in this change, and they're trying to actively streamline this biosimilar development. So first we had the revision of product-specific guidelines, insulins, heparins, where clinical efficacy studies were no longer necessary. But in more recent years, we've also had update of FDA guidelines, also the World Health Organization guidelines. We also had a very straightforward update of the MHRA guidelines. And more recently, in a very hot topic, has been the publication of a concept paper from the Biosimilar Medicines Working Party, which was talking about the tailored scientific approach. So this concept paper was published in February and has been open for public consultation until April. And in this time, the BNWP has been writing a revision of their guidelines, which will be available very soon. And here what they are talking about is the streamlining the need of waiving clinical efficacy studies in order to streamline more biosimilar development. And what this approach will do, what this tailored clinical approach will do, is to streamline this biosimilar development by reducing clinical comparability requirements if the previous steps, so analytical and functional characterization and PK, have shown adequate comparability. So basically, it allows to move away from clinical efficacy trials while maintaining the highest standards for biosimilarity. So now that these important changes are very near, I would like to navigate a little bit through the biosimilar pathway and talk about why these changes occurred, and also about the future challenges of the biosimilar regulatory pathway. So first of all, I would like to talk a little bit about the evolution of this pathway. And for this, we very briefly have to remember why the development process of a biosimilar originates from that of a reference product or an originator biologic. So here what we can see is the development process of an originator biologic, so a new molecule. And we have the size of each step representing the importance that it has in the approval process. So what we see here is that for an originator, a new biological, the main goal is to establish safety and efficacy in a specific indication and population. Therefore, stage 4, so clinical trials that prove the product's safety and efficacy in patients, is the most important step. However, when we're talking about biosimilars, these stages are inverted. Here, our main goal is not to demonstrate direct patient benefit, but to demonstrate high similarity with an already approved biologic. Therefore, the most critical steps will be steps 1 and step 2, where we are demonstrating, again, high analytical and functional similarity with the originator. And only if there are doubts regarding the comparability in these first steps will there be necessary to have a demonstration of comparability in further steps. Therefore, mostly clinical efficacy studies, which are in most cases confirmatory. So when the biosimilar regulatory pathway began in Europe more than 20 years ago, in 2004, there were many uncertainties regarding the translation of this analytical and functional similarity into the clinical level. Therefore, the initial regulatory framework had a conservative 5-step process that was very similar to originator biologics. A confirmatory clinical efficacy study was routinely required as a precautionary measure to ensure that the demonstrated similarity at the quality level was effectively translated into clinical similarity. After accumulating experience, it became more evident that non-clinical studies in animals did not add critical information, and therefore we passed into a 4-step regulatory pathway. But in more recent years, it has become more evident that these confirmatory clinical efficacy studies actually do not add critical information to the comparability exercise. And what has this evolution been based on? Why do we believe that this current 3-step process for the development of biosimilars is strong and robust? So this evolution is validated mainly by 3 drivers, in my opinion. First of all, there's been a huge regulatory experience gained in the past 20 years, especially in the European market, which was the first market. And just for a small piece of data, only in Europe, 126 biosimilars have already been reviewed. And that's a huge amount of experience accumulated in the regulatory assessment of biosimilars. Secondly, we have also had a great improvement of the analytical sciences and the techniques for the quality characterization, not only in biosimilars, but for all biological medicines. And last but not least, we have had different groups that have analyzed this information. So here we see in the slide that from 2020, there have been published several data-driven analyses by various authors, which include different products, different methodologies, different markets, and they consistently converge on the same conclusions. So for instance, I would like to go into the methodologies of all papers, but I have just summarized a little bit the conclusions. For instance, we had in 2022, Schiestel et al. They performed a retrospective review of 42 clinical trial results from PK and clinical efficacy studies of 38 development programs from EPARs, from the European market and the American market. And they found that in 100% of the cases, the biosimilar development programs, the clinical efficacy studies confirmed biosimilarity. And in 36 out of 38 cases, the clinical efficacy studies actually added no additional value to the scientific review process to approve a biosimilar. There were only 2 cases that involved biosimilar candidates that had differences in immunogenicity, but that, however, were not approved until the manufacturing process had been optimized to improve product quality. And these cases, the recurrence of these cases is very unlikely today due to the state of the art of the assays being improved controls of process-related impurities. Later on, we had again in 2022, Bielski et al. Also performed an in-depth review of 6 classes of monoclonal antibodies and fusion proteins, so complex biosimilars. And they also identified no instances where clinical efficacy studies added crucial information for the establishment of biosimilarity. Later on, we had Kirky et al., which also analyzed post-marketing surveillance data of 6 different monoclonal antibodies and fusion proteins. And again, what they saw is this post-marketing surveillance data that covered up to 7 years of follow-up, and more than 1 million patient treatments did not reveal any safety issues regarding biosimilars. And here we had these other 2 publications, which of course I'm a little biased when I'm talking about, because these 2 publications are the publications that were part of my PhD. But the truth is that these publications together have analyzed the largest set of biosimilars analyzed up to date. So they provide a data-driven analysis of the marketing authorization applications and the EPARs and the withdrawal assessment reports of all monoclonal antibodies and fusion proteins that were assessed by the EMA up to November 2022. So this was what this analysis showed us is that the quality package is predictive of the authorization of complex biosimilars, because there were no instances where the evaluation of the quality dossier and the outcome of this marketing authorization were not aligned. And what is most important, our observations showed that clinical efficacy studies did not play a critical role in regulatory approval. So uncertainties regarding the quality data were never resolved by clinical efficacy studies. And when there were uncertainties regarding clinical data, these were always resolved through the robustness of the quality package plus pharmacokinetic comparability in sufficiently powered studies, or justified based on the limitations of clinical data in biosimilar development programs, which means in mature secondary endpoints and balances in trial arms or chance findings, because it is already known that most of the clinical efficacy studies that are developed for biosimilars do not have potency enough for these endpoints. So basically, just to sum up a little bit, the conclusions from all of these authors from this data-driven analysis, which validate the evolution of the biosimilar regulatory pathway, is that clinical efficacy studies are no longer warranted by default for the demonstration of biosimilarity. And that's the other way around, that confirmatory clinical efficacy studies should only be carried out in exceptional situations where this data, this efficacy, safety, immunogenicity data actually provides crucial information for the approval of this biosimilar candidate. And what is most important for me that you get from this phrase is that this does not mean that less evidence is being generated for the demonstration of biosimilarity, but rather that the necessary evidence is being obtained through other means. So where are we going now with all these changes and with this evolution that is already happening? So as I mentioned at the beginning of this presentation, this evidence and the scientific knowledge that I have already pinpointed throughout the presentation is already being materialized worldwide. We not only have had also the update of many guidelines, this concept paper with an update of a new guideline for more complex biosimilars, but we have also had several multi-stakeholder initiatives, events, joint efforts that have taken place in the last years and that are focusing also on the specific topic of increasing efficiency of biosimilar development programs, like this webinar that we are hosting also today. But however, like all new changes and paradigms, there are also certain challenges that I believe that we are facing in this new paradigm. And I would like to talk about many things, but I think also for the seminar, I will keep it short. But I basically would divide them into two special challenges that I believe that are paramount, and it's important to take into consideration with the new types of development programs that are being done. First of all, is about maintaining the highest standards of scientific evidence. So when discussing biosimilar development without the requirement of clinical efficacy studies, it is very important to guarantee comparable safety and efficacy while maintaining the highest standards of evidence. So first of all, the comparability exercise at the quality level has to be particularly extensive, very carefully designed, and without any potential shortcuts. Also, we will have some products where there's probably maybe not such a clear link between the critical quality attributes and the clinical outcomes. And in these cases, sometimes appropriate studies may be necessary to increase this overall knowledge of the product's critical quality attributes. So in summary, it's paramount to ensure throughout the quality package that any observed differences that you find in the quality package will not impact the clinical performance and biosimilarity. And then another important issue or another important challenge regarding this new paradigm for biosimilar development is regarding the expectations for clinical data. So in this case, while a comparative clinical PK study is considered a basic requirement, the comparative efficacy study will probably not be necessary. Sufficient evidence on biosimilarity can be inferred from the other parts of this comparability exercise. So what's very important is that comparative PK studies should be performed in a sufficiently sensitive and homogeneous population, which in most cases will be healthy volunteers, like Stephan already said. But it's very important to take into consideration that this clinical safety and immunogenicity data that we get from this comparative PK study in combination with all the information that we got from the analytical and functional characterization will be the one that guides the decision on whether additional studies are required to show biosimilarity. This is also very important when designing your biosimilar package and your quality package and the design of your study in order to make sure, again, of what I said before, that any of these observed differences that you find in the quality characterization or even the PK studies do not impact or will not impact the clinical performance of your biosimilar. And with this, I'm reaching the final part of my presentation. I think I'm on time, but this is kind of a little bit the take-home message that I would like everyone to take from this presentation, and I hope that some people have gained some clarity with the data that I've been able to squeeze in 20 minutes. But in my opinion, biosimilar development is transforming, and this is driven by regulatory advancements, by scientific innovation, also by the analysis that have performed by the authors that I mentioned before. And shifting away from default clinical efficacy studies, we are now focusing on a very robust analytical comparability package and also pharmacokinetics in order to maintain these high biosimilar standards. And again, in this sense, it's very important for collaborative efforts and also for precise comparability exercises in order to address these future challenges and to ensure always that we have efficient and patient-centered progress. And that is all from my side. I'm not sure if I went very, I can't see you, but I hope that the presentation went smoothly.
Thank you very much, Elena. Perhaps one follow-up question. How do you foresee a potential effort to harmonize also on a global level between the EMA and the FDA?
This is a very good question. Well, there are already, so here there's also differences, I think, regarding even the definitions and the different jurisdictions and legal and jurisdictional issues, which in most cases does not have to do with the scientific issues, but with other things that are more complicated. But how I see the EMA and the FDA, I do think that they already are harmonizing, and I do think that they are already including more or less the same scientific principles. And I think, as many people say, so the heart of the people are in the right place. Many people are already do believe in the scientific evidence and this change, but there are always many issues regarding market and regarding legislation and jurisdiction that are hindering this process. But I do think that one way or another they will go in the same path, which is no longer default clinical efficacy studies, only exceptional situations, but always, as I mentioned before, having a very thorough quality package and a PK comparability study that is sufficiently powered.
Right. And perhaps one more question here, whether, not sure if that's too specific, but if the FDA is considering also the acceptance of non-US reference products in the PK/PD study, and likewise EMA, vice versa.
If I think this is going to happen, that's the question. If they will consider the acceptance. In my experience with the analysis of the products, I would say no. This has not happened in my experience with the analysis that we have done of these 33. So you had to have the European reference product and the US reference product, and you could not give the data only with you could not give the data with a US reference product for the marketing authorization application for EMA at EMA level. I am not sure this will happen. I personally don't think it will happen anytime soon. That's my personal opinion, but in my experience, this has not happened, and I don't think it will happen very soon.
Wait, one last one is coming in as we are. So the draft EU reflection paper is appreciated, but do you know if EMA has a timeline for when CES will formally be not needed, assuming analytics are robust? I think there is no definitive answers, but you know, perhaps you can share your viewpoint on what will actually now evolve soon.
Yeah, I can say that there's already a tool that can be used for this issue. So there's also the tailored scientific advice for biosimilar development, in which you can apply with the design of the development process of your biosimilar that you've had, and talk about how your quality package will be done, your PK studies, and already asked the question if whether a clinical efficacy study could be waived or not. And here I already do know that I think there have been some instances where in the scientific advice, again, I'm talking about scientific advice and I'm not talking about marketing authorization, where they have already said yes into waiving clinical efficacy study because the other parts of the comparability exercise were robust enough. So I think this is something that is already happening for some developers and something that is already on the table. Of course, again, this is always case by case, and it really depends on how you have designed the development of your biosimilar.
Great. So let's perhaps park then the remaining questions for the end of our session, and otherwise we'll also be able to address them offline. Thank you so very much, Elena. Really fantastic insights. And now, Martin, if you can share your presentations, welcome. So again, Martin heads up the biotech division at Midas Pharma, a company that we collaborate with in various projects. And yeah, happy to have you here today.
Yeah, thanks, Stephan, and thanks also to the Conscio Group for giving me really the opportunity and the chance to speak here in the webinar and to share my experience from the past And with Midas and from my previous life on common pitfalls on the way from R&D to commercialization of a biotech product. And I'm really looking forward to the next 20 minutes to show you my experience. But before we do that, I want to quickly introduce Midas and what we're doing. We were founded in 1988 by Karl-Heinz Schleicher. He was in the C-level of Boehringer Ingelheim and then founded Midas to establish a fully transparent purchasing organization for small molecules. Developed now the company. In the meanwhile, we're doing 550 million on turnover annually in 60 countries. We have around 350 employees in 10 countries located across the world. And we're, besides the core business we did initially on small molecules, we in the last few years decided to step into the biotech business and act as a service provider for the biotech industry and help young startup companies and also bigger pharma companies, mainly from the Asian market but also internationally to bring their products to the European market. And we work closely with the Conscio Group on some of the topics here. Yeah, as said, we're located on 10 countries around the world. We have a huge presence in China. We have a huge presence in the US, where we also have a regulatory staff to help supporting BLA filings with the FDA, for example. We do have a presence in the BioHub in Basel in Switzerland to be connected to startups and be at the heart of development in the biotech industry. At the moment, we're dealing with 8 commercial products, and we're acting as a service provider of one of the biggest biosimilar companies in the world, acting as a batch releasing site into Europe for their products, and also acting as a MAH on some biosimilar products for companies globally. We act along the full value chain of biological products, starting from early cell line development towards the whole product and process life cycle through the commercialization. And that's where I want to start and dive into the topic, where I see the common pitfalls that could happen on that way. And from my perspective, we have 5 major cornerstones where commonly pitfalls could occur and which could tremendously delay or even lead to declining of your product application when you step or fall into these pitfalls and you do not consider them upfront and do a well planning to avoid those pitfalls. This is first in regulatory compliance and data quality. I think that leads into what Elena said already in her presentation, is financial planning, is IP management, is strategic partnerships, which is specifically interesting and necessary for small startup companies, and then the market assessment. So going into that, and I want to start with the regulatory compliance and data quality. I think also, as mentioned by Elena, it's super important to engage early with your regulatory agencies through scientific advices. It's super important to be transparent here, super important to be open and to have a clear presentation of your briefing books to the agency, because I saw a lot of times that people and companies sometimes not want to disclose let's say quality topics, or they even not sure about quality topics at the point when they file briefing books and say, no, we not want to disclose this issue now. Let's see if it's later comes up in the review, then we can clarify it there. I think that's not the right point. I think the right point is really upfront, be open, be honest, be transparent, and seek the way and the advice from the agency to partner up. And then typically you have throughout that meeting notes and feedback from the agency that helps you and ensures a smoother review process and approval process of your biological license application or MAA filing in Europe afterwards. One integral part of that is data quality. I think you absolutely need to be aware of your process. You need to be aware of your product quality. You need to define properly upfront critical process parameters through your development lifecycle. You need to define critical quality attributes in a proper way in order to characterize the product properly. And for me, key is to use quality by design as a key advantage in your process development. Yeah, as said, you just need to know your product, the process, and the data in order to convince also agencies, for example, if they come for inspections, because that's one of the pitfalls as well. What normally happens is you go into the commercial manufacturing phase. So you filed your application, you're in the review process, reviews are answered, and then the last hurdle in order to get approval is eventually, if you're not a European company, the inspection by EMA to get a EU GMP license, or for example, a pre-approval inspection from US FDA if you're a company located in outside the US and you want to get on the market and not hold a US GMP certificate. So, and that last hurdle could always be influenced if you're not meeting the compliance requests and not be on spot with data integrity requirements, with GMP requirements from the local agencies. Specifically with US FDA, that's a pain point, I can tell you. But also it's a pain point if you're not considered that such agencies like the FDA are not focusing and spot checking you at your actual process and the GMP compliance, but also could go back into your development data during the inspection and check on the development data and data integrity on your development data to see and verify if the claims you made in the R&D section of your initial BLA filing are accurate. And that's what they're actually doing. And then in the past, we saw several occasions where exactly this happened, that they went back into the R&D data, challenged the R&D data, and had observations through a pre-approval inspection in site at those R&D data. And this can be easily avoidable in sticking to the compliance from the beginning, also in R&D, where most of the companies not necessarily think towards commercial manufacturing and GMP compliance, because they say we not need that because we're doing early stage R&D, or we not need data integrity. Okay, if we look into IP management, I think we need to differentiate a little bit if you're a biosimilar manufacturer or you're an originator. I think for an originator, it's absolute mandatory and integral part of the development process to develop a robust IP strategy, how to protect your product, how to position you on the market and how to enhance market position, and how to secure your competitive advantage through this IP landscape and securing your knowledge throughout the years and avoid that competitors come into the market. Now, if you're a biosimilar company and try to develop biosimilars, it's I think important to know the IP landscape, to not violate against any IPs, because that could be a huge setback in your strategy when you all of a sudden find yourself in IP litigations with your competitors or with the originators. Yeah, we saw that in the past, for example, that clinical material got blocked in customs because there were originator claims of IP infringements, and that set the companies back 1, 2, 3 months in their clinical trials because they could not supply clinics and supply the clinical trials with material because it was stuck in custom. Or even I saw one instance in one of my previous companies where we developed a fusion protein biosimilar, and all of a sudden we were under IP claims. I mean, that was back in 2015, I think, and we find ourselves instantly in a court trial where we were forced and requested to destroy everything that was manufactured up to that time point, which was like R&D material, clinical material on stock, cell clones, cell banks, because they claim this is an IP infringement that you start development of such process. And well, we were lucky. We had a site outside of the IP landscape where it was legal and eligible to hold that product. And luckily, part of the development work was there. Cell banks were there, so we were able to reconnect and plug it in after having that major setback. But that's something in future you could absolutely avoid in doing proper market assessments. What also is really important, I think, is doing a licensing and IP enforcement, negotiate with your competitors, negotiate with the originators to find settlements with regard to the market entry. And make sure you have a proper launch phase support, enough material to support launch and be ready to supply the market. Now, if we look into market assessment and financial planning, I think it's key really to know the customers. You need to know the pricing strategy for your customers. Are you supplying into hospital, into retail? How is the prescription area landscape? How is the reimbursement strategy for the product? Because that's really key for you later onwards to be fast and not really connected to launching, but that's your enabler to be fast in the market and to act quickly and be ahead of your competitors. And you can use that as a commercial advantage, so to say, to bring your product quickly to the market. What we always recommend to do our customers, and we do that with our in-house team here as well, we help them to do a competitive landscape analysis to know the competitors that are on the way to the market as well. We try to seek and get their pricing strategy to plan ahead and be better or different. And we upfront always encourage our customers to plan their market share appropriately. And then it's key essential, as said before, do launch support, do stockpiling batches, have enough material available, and be so well financed to be able to supply the market, not only for a launch, but also then for continuous supply, because that's what I see in the past as well, specifically with small companies, that they are not that well funded and then struggle to supply a super high price product into the market because they're not able and not have enough money to manufacture a commercial product and keep demands high enough. For me, it's also important to have strategic partnerships, and that's actually the main focus area me and my team are working on, because such collaboration with an established firm, and it's especially important, I think, for smaller companies when you're not able or you not want to keep a huge and fund a huge crowd of quality people, regulatory affairs people, sales force. So partnering up with people helps you to enhance your credibility. You can widen your manufacturing network. You must eventually not focus on getting high price equipment to manufacture your product, but you can rely on partners to do that. You can leverage services like QP services, PV services, batch release activities, import-export service, those things. That could lead definitely to a cost reduction at your end, because you not need to keep up that high level in staff costs. You share the risk eventually with a partner. If you partner up for marketing and sales, for example, or you out-license your product for wholesale and distribution, you can leverage the sales force of your partner, and you just can create and use synergies throughout such a partnership. Yeah, having that said, Thank you very much. I think it was a little bit faster than the 20 minutes. Thanks also to my team, who's really doing a tremendous job helping and serving our partners on a daily basis and enable them to bring great products to the market.
No, particular questions right now following the presentation. So I suggest that we keep the rest then for.
I think I saw one. There is one from Shoba, I think, says, what aspects of research and development data? Oh, well, maybe it's not specific to Martin's presentation.
Okay, I don't know, Martin, can you see this? What aspects of R&D data that go into the dossier are generally audited by the US FDA? Which studies conducted in R&D are generally audited by US FDA? Is CVR 21 compliance required for R&D data?
Yes, yes, and absolutely, I can answer that. It's not required, but they definitely and typically they not audit you on the clinical data. As said before, their main goal is to come in your facility and check the GMP compliance and adherence to 21 Annex 11 and those CFRs, 21 CFRs guidances in general, so the general GMP compliance. But what they and all the clinical data, so to say, are reviewed during the scientific review and discussed there, and absolutely you not need to follow the Annex 11 requirements for all your early stage R&D data. But what they definitely do, and what they could do, and they fully have the right to do, they can go back into your R&D data and see if your R&D data that were filed with the dossier comply with the actual primary raw data that you have at hand. And if they have then any hint or indication on data integrity, for example, that you create the impression that there's data fraud, that there's data manipulation, that there's active deletion of data, such activities, I think then you ultimately will be asked how you ensure your data integrity and also eventually 21 CFR Part 11 compliance throughout your R&D phases, and that simply could be that you want to provide them or are requested to provide your data governance strategies for your R&D labs. Yeah.
Excellent. Thank you very much, Martin. And over to you, Clemens, on bioassays.
Thank you very much. I'll start with the sharing. Just give me a quick information when you can see my slides. Still loading?
Yes.
See there? Perfect. Awesome. So yeah, thank you very much for having me here in this webinar. Right now it's getting a little more technical. I try not to bore you out with details. I don't have any like complex formulas in here, so don't worry. But when we set up this webinar, we thought about what makes sense and where we're talking a lot about this time to market here and where we can speed up the process. And as you will see, we know that the analytical data is very important. That's also Elena already and also Martin pointed this out in this biosimilar approach. And then, as you will see, there is the bioassay especially has a lot of potential for speeding up the process, and that's why we included it in this webinar series here as the final talk now. Just a few words to me. So I'm right now working for Conscio Pharma Austria in Eisenstadt. Currently in my role as quality assurance, but I do have quite a few years of experience in the cell culture lab actually. By doing validation of bioassays like from scratch up, and also doing transfer validation, development, troubleshooting, and also a lot of experience with biosimilar release. So I hope I can give you some insight here. So let's now try to come from this rather general view that we had in Elena's and Martin's talks down to where we are now talking about the bioassay. So for our assay itself, it's all about establishing biosimilarity. And here, if you compare the EMA guidelines and the FDA guidelines, they are pretty similar. And what you can see, and also Elena pointed this out already very nicely, the analytical studies of establishing this biosimilarity do have quite a lot of importance. So the better you are prepared with your analytical studies, the more likely you are in waiving some of the clinical parts then in the end. So I think it do make sense to actually put some effort in here, develop assays that can then actually are state of the art and can then be used to waive a little bit of the effort then in the end. So if we if we now, let's quickly get an overview on this analytical studies. So what is typically my release portfolio for a biosimilar? It is mostly similar to what you would have for an original product as well. So you would have your biophysical properties that you have to study, like composition, physical properties. These are mostly like HPLC methods, post-translational modifications, which are very interesting, of course, because we are working with a biopharmaceutical. So we have an expression host where our protein is usually expressed. So these have a very important role during your analytical studies. Then, of course, potential variations that happen either during the expression of your protein or then later on, deaminations, oxidations. Of course, also intentional modifications like your petulation sites, if this is relevant. Then, of course, a huge part and also an important part are your immunochemical properties, target affinity, FC receptor binding, FAP and FC associated effector functions. Then, of course, similar to your originator, it's like purity and impurities. Your impurity profile, it could be different to your originator, but you need to show that your impurity profile basically also fulfills what you would have for an original drug as well. And then last but not least, it's the biological activity, a very important assay. You have basically 3 different methods available still, but actually only 2 of them are still used. So it's on the one hand, it's your ligand or receptor binding assay, and we will come to this in a second. Then it's your cell-based assay. This is what this talk will mostly be about. And then lastly, the functional assay, so where you would test your drug actually in animals, but not for toxicity, but actually for showing biological activity. This is fortunately no longer part of today's release portfolio, because as we all know here, it's not necessary to have animals here. The variation in those assays is very high, and actually the scientific knowledge you gain with these assays is rather low. So unfortunately, those are no longer actually used. So we are left basically with the ligand and receptor binding assay and the cell-based potency assay. So just quick from the guideline, biological activity, it describes the specific ability of a product to achieve a defined biological effect. And if you talk about biological activity in those kind of assays, we usually call it potency. So it's basically the quantitative measure of the biological activity. And in the sense of biosimilarity, you will see that this assay, the potency assay, has very high importance. So a quick, just to get everything on board, I don't know like what your backgrounds are exactly. So let's just quickly summarize how will such a cell-based potency assay look like. So usually you have your cell line preparation, you seed the cell line, could be adherent cell lines, could be suspension cell line. Then you treat them with a serial dilution of your biopharmaceutical, have usually some incubation steps, maybe like treatment is not just 1 step, could also be 2 or 3 steps. And then in the end, we have a measurement of our endpoint, and we produce, as you can see on the right, those nice 4PL curves, so 4-parameter logistic curves. Here it gets a little bit technical, but bear with me, and then we're done with this. 4PL, what does it stand for? 4 parameters. So the curve on the right, this S-shaped curve, you can see it has a bottom, that's 1 parameter. It has a top. I'll try to, show my, where is it? Yeah, there we go. So we have basically it's bottom of the curve, so where there is no decrease of signal with a decrease of concentration. We have the top of our curve, so where basically an increase of drug concentration does no longer lead to an increase in signal. Then we have the slope, which is the B parameter, or actually it's not the slope. Let's call it parameter B because it's not really the slope, but it's to simplify it, it can be explained with the slope. And then we have a very important parameter, and this is the EC50. So basically the value where half of my signal is reached. So in this case, it's roughly around here. And then potency, by comparing, it's in most cases we call it relative potency. So we compare a reference, an originator, for example, to our biosimilar. We would expect both of those curves to have the same top, the same bottom, the same slope, and only differ in EC50. And this shift in EC50, this is what then we would call relative potency. So if you would need more drug to achieve the same effect, then we could say, well, there's probably a difference between my reference, between my originator and my biosimilar, because I would expect to need the same amount of drug to reach the same effect. Pretty straightforward. And now also one important point that you can see here already is it's a cell-based assay. We are working with bioassay, we're working with a living organism. And you can see here in these dots here, always the triplicates in this case of my data set. You can see here that those data varies quite a lot. So this is not what we would expect in variation if you just do a simple ELISA or something, but that's part of an bioassay. We do have variation. But we will come to this also during the talk, whether or not we should accept this variation and should accept the fact that, yes, bioassays are inherently have a high variation, or whether or not it might make sense to reduce this variation and tackle it in order to improve the bioassay. So real quick, as promised, the more simple form for this assay, so the simple receptor or ligand binding assay, would be the ELISA, as most of us know it. So we have our biopharmaceutical, we have our drug. In most cases, we are talking about antibodies, and we can simply look at the binding of my antibody, of my biopharmaceutical to my target, which is in most cases a receptor or another ligand. And we simply look at this binding, and this tells us already a lot about the higher order structure of my protein, because if the binding works, then most likely my protein is not misfolded. And that's basically also the authorities' view. Why are we doing these bioassays? Because we have a lot, as I discussed in the previous slides, we have a lot of other assays, and those assays are improving our, like we have mass spectrometry. We can even, like today, very nicely look at our protein, but still the authorities want to see this so-called mode of action assay. They want to see that you have an assay where you can actually say it's not just the same from an amino acid sequence. It doesn't have like just the same properties physicochemically, but it actually also binds to my receptor in the same way, because this is basically the ultimate confirmation that, yes, very likely you have been producing the same protein than the originator. So that's the receptor ligand binding assay. The advantage of these is, of course, very low assay variation. If you think about an ELISA, you have a lot of platforms available. You have a lot of kits already available. They are usually high throughput. You don't have any like cell culture to prepare first. But the drawback is, of course, less clinical relevance, because you're just looking at this first step that your biopharmaceutical does in the body. It's just the binding to the receptor. You're not looking at the downstream signaling or anything else. So what usually is the only accepted option that you have, if you're talking about release portfolio, is the cell-based potency assay, that you have a cell line where you can actually mimic and reproduce the mode of action of your drug. So you pick a cell line that is usually, if you have a drug against breast cancer, you usually pick a breast cancer cell line that has the receptors that you are looking for, and that also then in the end gives you the final result that your drug would also elucidate in the human body, in the physiological one. The advantage of those cell-based potency assays is you have the ability to reproduce complex mode of actions in your cell-based potency assay. They have increased clinical relevance because you're not just looking at this first step, not just at the binding. You're actually looking whether or not the downstream signaling is also induced, and whether or not the effect in your cell line is reached. So way more clinical relevance, and I think Elena pointed this out really nicely. It's all about the clinical relevance in your analytical data set. And as we said, the cell-based potency assay allows us to really infer the higher order structure of our proteins, because if we can show in the cell-based model that our proteins, our biopharmaceutical is doing the same thing, the likability is very high that we have actually produced something that is biosimilar to what we are trying to do to our originator. So the drawback of this is, of course, sorry. The drawback of this is, of course, the assay procedures are more complex. So we have this living organism involved, we have our cell culture involved, so the assay usually takes longer. And all of this usually leads to an increased assay variance and to a decrease in accuracy, especially compared to a simple ligand binding assay. Here's just a quick example of what you can actually build or do with the cell-based potency assay. So in this case, we're looking at rituximab, at this drug, and rituximab in this case has not just one mode of action, so rituximab induces more, so it works on one hand in CDC, so complement-dependent cytotoxicity. That's one way how it's working. Then we have ADCC, antibody-dependent cellular cytotoxicity, and so you can see it's like you have this possibility with the cell-based potency assay to reproduce even more complex mode of actions and show biosimilarity. Now that we have covered basically the basis, let's see where we can actually save time, where it actually makes sense to put in time. If you're now thinking about development and validation of your bioassay, at one point you most likely will transfer your bioassay out to a contract laboratory. So in most cases, if you start with biosimilar development, in the initial stages, you will probably also have your bioassay in-house. Maybe it's at that stage still a receptor ligand binding assay, or later on the bioassay, because you might also need the bioassay during clone screening or whatever. But at one point you will outsource it, so you will have to do a method transfer. And as it's put here, the successful method transfer starts already at assay development. So you want to have robust and bioassay with a low variation, because this will then also make the transfer way easier, because all of the variation that you already have in-house only multiplies by a certain factor if you try to move this assay to another laboratory. So one thing is pick a robust assay format. And here I will talk about reporter gene assays at a later stage again. Think about choosing reporter gene assays versus the traditional cell-based bioassay. I will then explain what I mean by this. But the first thing is actually choose an assay format that inherently has less variation, which is kind of a no-brainer. And then it's about your cell lines. Of course, you need to establish a stable and well-characterized cell line and cell banks. So yeah, think about minimum, maximum passage number. Know at what passage number your cells actually work in the assay. Have a growth curve. Have the ideal medium compositions figured out. And these are most likely not the medium compositions that are shipped, that the cell lines is shipped with. So it's possible that you need to play around with here a little bit. Think about splitting schemes, how you do a routine cell culture, how you prepare your cell line before it goes into the assay. Think about the incubation conditions. Those are also very, very important point. And then try to minimize assay variation as far as possible. So as far as it also makes sense from an economical point. So do not need to go down to low percentage in confidence interval for our potency estimate. It's still a bioassay, but you should also not be happy with having like a potency estimate and a confidence interval for this, which is basically the variation of how confident I am with my estimate, that is above 20%. And we did see this, so we did have confidence intervals between 20 and 30%. That's probably not an assay that we need to show biosimilarity. It might work for an originator, because there it's basically, yeah, the mode of action is you can show it, but now we want to actually show biosimilarity between two products. So we want to have low assay variation to are confident in our results. And what the point that you hear a lot in this context is always either validate or standardize your assay variations. And I would usually go with the standardize first approach, and everything that you cannot standardize, you can still validate, and then have basically in your robustness part of your assay, and that you know during the transfer, these are parameters that you can play around with, that you can change, But you also know about the parameters that you should not change. They're absolutely to be standardized, and this makes transfer them a lot easier. So this is not just like a rough overview. Please do not like nail me down on the numbers. Like he said, well, the reagent temperature makes up 5% of my assay variation. It's just to give you a picture. You do have a lot of sources for variations, and also sources that you would usually not think about. And those all add up. And if we have those sources for variation under control, and basically that's what they see with the comparison between not standardized and standardized, then the only thing that we cannot control here are our cells. We cannot control the inherent variation that comes from the cells. But if we think about bioassay, we usually think, well, all my 20 to 25% of assay variation are all from the cells. I cannot do anything about it. And that's not the case. There's actually a lot of variation that we bring into the assay during development that we might not be aware of. And that's why I listed them here, a few of them. So reagent temperature might be one of those where I say, yeah, of course, it's a biological assay. We're talking about enzyme assays usually, or protein interactions. Of course, temperature makes a difference. But then it's also cell counting. It's also cell detachment. So already how I prepare my cells, what I usually would not consider to be part of the assays, like it's preparation, and then everything else is my assay. Also, cell preparation is already part of your assay. And there are a lot of different ways on how you can do cell detachment. And I'm teaching a cell culture laboratory also by the side. So I know there are like a lot of different ways how you can detach your cells, and you can already kill your cells during the cell detachment, or at least bring down the cell viability to a point where you have assay variation that is not necessarily needed to be there. Shear forces is another point. We had an assay where it actually had an influence how often our cells had been resuspended before we bring them to the assay plate. Because we do introduce, of course, a lot of shear force. It's like we're working with a living organism. It's not just putting reagents together. There are cells, and we should treat them as standardized as possible in order to get the best result. And if you're, also this is an important point here, standardized as possible does not always mean the best situation for the cell. So if you think about, for example, medium temperature, you're doing your cell preparation, you put out your cell culture medium, and we usually would put it in a water bath to get it up to 37 degrees because that's the coast temperature for our cells. And then we take out our cells and we start with the working. As soon as you take out the medium from the water bath, it will drop down to room temperature again. But it will not drop down immediately. You will have a steady decrease of temperature of your medium. And now you have, again, a source of variation, because depending on how fast your operators are working, they will be working with differently warm cell culture medium. And it might now sound to you like, well, how that doesn't make a difference? Yes, it does. So in this case, it would be better to not bring the medium into 37 degrees at all, just bring it to room temperature, because your cells will not die with medium at room temperature, but you will have one variable that is fixed suddenly. It's not the best probably for the cells, so maybe like 2 or 3 cells more would survive if the medium is warmer. However, your assay is standardized. So these are things that you should think about on how to reduce these sources of variations, to reduce these error sources. Incubator is also a very important point, and I will discuss this in a second as well. So one point is always, if we have too high assay variation, we can either, that's what I would say is the better way, is decrease variation, or what is usually done is we simply increase the number of plates. We increase the number of individual measurements that we do, and then we can combine those, and then of course our variation will go down as well. And this is usually the standard approach. So we see a high assay variation, we say, well, okay, let's just increase the number of plates. Let's just do 2 measurements, 4 measurements. We had assays that had been transferred to our contract laboratory where one point estimate needed 6 individual assay plates, like 6 complete individual approaches, and then you would be able to combine those measurements in order to get a confidence interval that was low enough and good enough. So this is one way it could be done. There is also just one thing to be said about the combination of the assay results, is that usually what happens in a lot of cases is that people take those independent potency results and then they compute the mean and then the relative coefficient of variation. And this is just one thing. This is scientifically not correct. So the mean is of course correct as your potency estimate. However, do not take simply the 5% coefficient of variation because it does not translate to the actual confidence interval of your potency estimate. There are basically 3 different options available, and most potency evaluation software support all of them. So it's basically either an unweighted combination where all potency results are treated the same. Then you can have an homogeneously weighted combination where basically the weight is calculated for each potency value individually based on its confidence interval, or you can also have a heterogeneously weighted combination. So this is just as in for you. I think this might go a little bit too much into details, but let's now go back and see where can we actually decrease variation. So what are actually hands-on examples on where we might increase or where we might introduce sources for variation for our assay that can easily be removed. And one of them is have a separate incubator for routine cell culture and assays. We saw it often in laboratories that there was just one incubator, maybe a second one for as a backup. And in this incubator, routine cell culture was performed next to your assays. And in the incubator, you have ideally 37 degrees, in most of the cases 5% CO2, but each door opening reduces the temperature, and of course also you have a drop in CO2 level. We did this at the laboratory where I worked with. We monitored the CO2 incubator, and during this work day, I mean, there are students in there, there's of course a lot more traffic in the incubator, but still the CO2 level never reached the 5%. So during this continuous opening of your door, your CO2 level never goes back to where it should be. But you have an assay in there where you want to have as little variation, environmentally speaking, as possible. So one thing would be simply get another incubator that is just for assays, that has little to no door openings during the incubation of your assay. And thereby you have removed one very big source of variation. And also, if you get one, it's possible to get them with segmented doors so that you do not have to open the inner door completely. As you can see in this image here, you have basically like 6 segmented doors, and thereby you even further reduce this drop in temperature and CO2. Medium, also a very important point, our cell culture medium. One thing that is very critical that we saw as in our experience during bioassay validation is the FCS, fetal calf serum, which is in basically all cell culture mediums because it has a lot of growth factors in there, it has a lot of cytokines, a lot of stuff, but it's not defined at all. Fetal calf serum is basically like, yeah, it's from fetal calf blood, basically. It's harvested, and then you have like huge lots which are roughly similar, but those lots usually expire after 3 to 5 years, and then you have a change in lot. You need to buy a new lot, and you get a different product because fetal calf serum is not defined at all. So it's basically like a natural ingredient. So it has a lot of variation, and it also has a lot of influence on your assays. So what you should always aim for, although it's a little more expensive, but still go for serum-free mediums. So choose medium compositions where the serum, the FCS, has been replaced by defined additions that you add to the medium. So only add the growth factors to your medium that you actually need for your cells. Only add the hormones that are needed, only the cytokines, whatever, that actually your cell line needs. And thereby you save all those money on those lot-to-lot changes, because after 3 to 5 years, either you or your contract laboratory will inform you that the FCS lot has expired and that a new lot is coming. And then you have this lot-to-lot change, which is expensive, and a lot of people forget about this during the initial development. So switch to serum-free medium in the first place. Is a little bit expensive in the beginning, but saves money and also time then on the long run. And then we have 2 more steps where we saw in our experience that a lot of variation could be introduced. It's correct pipetting, and this, I know we all think, especially if we're working in laboratory, how hard can pipetting be? It's the stuff that I do all day, so it's basically like driving a bicycle. I can't do anything wrong. What we performed in the first company I was working for, we performed basically a serial dilution. So we had like bovine serum albumin, a protein solution, and we had all of our operators perform this serial dilution in the same way. They got the same basically instructions, and then they put out the serial dilution on a 96-well plate, And then we measured the absorbance basically. So we just measured the protein concentrations, plotted those lines, and then compared our operators to each other. And you would not expect the amount of variation that you have, that you can produce there. And every one of us was thinking they were working correctly, and all of them had a lot of experience, but you get a little slaggy, a little, yeah. Everyone introduces their own errors in pipetting. And then we had a training. We had actually an external trainer come in-house and remind us of all these correct pipetting techniques, and then we repeated the stuff, and the variation dropped significantly. So that's also one thing. You would not think that your pipetting can introduce like a few percent of variation, but it actually can. So it makes sense to look at this. And then lastly, it's also, of course, cell handling. And what we saw here is that usually we would split our cells. If you are familiar with cell culture, you usually say, I let my cell grow in the dish, and once confluence is reached, so once all the whole surface is covered with cells, I just take the plate and I split them. And this splitting comes from the fact that usually you would say, well, I split them in a 1:2 ratio, a 1:4 ratio. So take all the cells, and I take a fourth of those cells and put them on a fresh plate, and then I wait until the plate is confluent again. That's the usual way, the standard way. The thing is, this leads to very high variation in cell densities, because at your assay day, it's possible that your cells are maybe a little more confluent, that they are already over-confluent, and if cells get too confluent, then they usually, first thing that they do is that they change the expression of the receptors, so they change basically the reception of the expression, of the very receptor that you need in your assay. That is basically the first step for your assay outcome. So you already go into your assay with different cells. So what makes more sense is to split your cells by seeding a fixed cell number. So don't say I take fourth of this plate and put it on the next plate. Say I always seed 100,000 cells for a T75 plate, for example. And again, you reduced one source of variation. Resuspension we talked about. So resuspension introduces a lot of shear force. So have all your cell culture operators trained in a way that they do the resuspension in a similar way. And also cell counting. Cell counting can introduce a lot of variation. And this is the case for both manual as well as automated cell counters. So even with an automated cell counter, you would think, well, it's automated cell counting, the operator is removed. No, it's not. There's a lot of preparation that you still need to do before you put your slide into your automated cell counter. So also there, you can remove a lot of variation. Yeah, heading to the final slides here, hope to stay in time. There's just one thing that I would talk about is like best practices during bioassay transfer, because we said at one point you will need to transfer your bioassay, and this means communication, and also Martin already had this topic as well. It's a lot about the communication between different stakeholders. In this case, it's about the communication between you or another contract laboratory that you already have that has to transfer the bioassay to a third contract laboratory. So what is important here? And in this case, I would say it's the involvement of the operators from both laboratories early on. So you do not want to have supervisors talking to each other about the assay. You actually want to have the operator that does it in the sending laboratory directly talk to the operator that does in the receiving laboratory on a very like basic level. And ideally, you do have at least 1 or 2 on-site visits. So in our experience, it is almost impossible to transfer a bioassay to another laboratory by simply sending them the SOP and maybe the reagents. They will not produce the same results. They will not do the same bioassay. So it's very crucial to have, I know I'm working in quality assurance, so I know ideally our SOPs are written in a way that everybody on the planet can reproduce them, and there's nothing that is not written in the SOP, but that's not the case. That's not the actual case. So you do want to have an operator from the sending laboratory in the receiving laboratory, looking at what they are doing and giving them tips or hints. And you might even find out stuff that you then later on want to add in the SOP, because simply the sending laboratory has not thought about it, or has always been done this way. But have them on-site visits, and have those on-site visits early on, because we also had transferred where we spent 2 or 3 months trying to figure out how they did the bioassay before they then finally had their first on-site visit in our laboratory. And then you figure out, well, okay, what was a simple handling step that could have been prevented early on. One thing we also had very good experience with is by simply ship your working cell bank to the receiving laboratory. So it's always better you will have a working cell bank prepared anyway. It always makes more sense to send them the working cell bank and the receiving laboratory starts working with them compared to them establishing their work cell bank on their own from your master cell bank, because again, a lot of source for variation and shipping a working cell bank is no big deal and can easily be done. Of course, equipment should be compared, so make sure that you actually have the same equipment, that the equipment specifications are the same or similar, that you can actually reproduce the assay. And yeah, as we said in the beginning already, try to reduce the assay to assay variation in the sending laboratory. So try to minimize the variation already before you send away to bioassay, because as we said, those initial variation is simply factored is multiplied by a certain factor once you try to reproduce it in another laboratory. And finally, what I wanted to say here, it's one point to make, is basically the use of reporter gene assays. So if you are familiar with cell-based bioassays, in the past, usually we had like this, let's call it traditional cell-based bioassay, where we would pick a cell line, treat it with our biopharmaceutical, and then wait until the very end point. So in this image here, we would not just have the receptor binding, we would also have, of course, the downstream signaling cascade that leads then basically to the upregulation of certain proteins. And then we would wait for the actual phenotype. For example, in TF1 cells, we would wait for the differentiation of those cells and then measure this. Now, we have seen with a lot of companies when they are going into the biosimilar market or they're producing a biosimilar, that they simply copy the cell-based bioassay that has been used for the originator. And when we then suggested to them the use of a reporter gene assay, then they always said, well, yeah, but it's like the originator has a different assay, and this is okay, but the reporter gene assay might make more sense. What's the difference now? Reporter gene assay compared to the traditional assay. It's basically you have the same binding, you have the same receptor, your biopharmaceutical binds. You also have the complete activation of your downstream signaling cascade. So you can actually show the biopharmaceutical is not just binding, it's actually leading to receptor phosphorylation and all this stuff. All the intracellular downstream signal events do happen, all the way down to the upregulation of certain proteins. But in our case, we're not upregulating the proteins that then lead to this phenotype change in our cell, but we're upregulating in this case it's luciferase, it's in most cases, so that we can add a substrate and have a light emission. So we basically at this point, after we know that the signal cascade has been activated, we actually get a direct feedback in light emission, and this is directly proportional to the amount of receptor that has bound here. So yes, it's true, we are like capping away a little bit of the original mode of action, if you want to put it like this. But our assay variance is decreased tremendously. Our assay time is decreased by, like, traditional assay can take 1 to 2 weeks. Reporter gene assay usually done in 2 or 3 days. So you can actually, and by reducing the assay variation, that's the point I want to make here, you actually are able to compare a biosimilar to an originator. Because if you have a traditional bioassay and a variation or confidence interval of 20%, then your originator and your biosimilar will always be basically the same, simply because your variation is so big that it's all lost in this variation here. But if you have an assay that has very low confidence interval, very confident point estimates for your potency, you can actually see difference between originator and biosimilar. And this is also one thing that Martin already pointed out. You can suggest those assays during those scientific advice meetings. And we also had this experience. During the scientific advice meetings, you can go down to this level. You can suggest, you can say, well, that's the traditional assay, that's their release portfolio. We are thinking about this assay. We have preliminary data. Let's do this. Yeah, Martin, do you have a question or?
No, I just wanted to add, I mean, you even see in some instances, and I saw it in the past, that companies are starting to take SPR assays, for example, biochar assays, to remove the classical bioassay because the SPR is not robust and but you just, as Klemens said, you need to well define and be ready to explain why you think your chosen assay is comparable or even superior of the assay provided by the originator. That's
yeah,
the best.
thank you. Yeah. Exactly. Yeah, and this is also then brings me to my last slide. We already have biosimilars on the market that have been purely released on reporter gene assays. So these are just an example. These are just 3 biosimilars where the release assay, the mode of action assay, was a reporter gene assay, and there is no, like, it's different to the traditional bioassay of the originator, but the data you get is gives you more confidence, and that's why it makes sense. And this is just one point. You should not be afraid of going this reporter gene assay way. That's what I want to say here. So thanks. I hope I kept my time. Thank you for your attention, and yeah, I'm open to your questions.
Thank you very much, Klemens. And again, to all of you, there's I think one question that might be more directed to Elena. Thanks for all the presentations, given the increasing importance of advanced analytical and PK/PD studies in biosimilar development. Under what specific circumstances or for which types of compounds do you believe comparative clinical efficacy and safety trials are still absolutely necessary? Are there particular characteristics of the molecule or clinical context that make these trials indispensable? I think a very, very valid and very good question, perhaps for Elena.
Yeah, the question that everyone wants the answer to, right? Well, I do have my guesses on specific scenarios where I do think that I don't think that there were always be absolutely necessary. So all the extremes, I think, as science evolves, this will always also evolve. But I think that right now, so in the current framework where we are right now, I do think that clinical evidence and most surely clinical efficacy studies, so studies in patients, phase 3 studies, will be necessary for very complex molecules for which the mechanism of action is not completely known or very difficult to characterize, because as we all have mentioned, so Martin, Klaus, all of us have mentioned, if you need to really understand your reference product and to be able to characterize it fully on a quality level, to be able to weigh the clinical efficacy study. So you need to understand what the differences in the quality attributes mean as into clinical impact, et cetera, et cetera. So for those molecules in which we cannot understand the mechanism of action entirely or characterize that, then this will just not be possible. And when we're talking about complex molecules, we're talking about, well, for instance, ADCs, we're talking about ATMPs, we're talking about all those molecules where we do not fully understand how they function in the organism, and well, we will always need some type of clinical evidence. Also, I would say situations where we have molecules that have that are very highly heterogenic or very difficult to characterize in their structure. So we can have, for instance, biologics that have a lot of structural heterogeneity, so heavy glycosylated proteins or enzymes with the complex profiles of oligosaccharides, et cetera, et cetera. And then also, I think that another important area right now will be those situations where PK studies are just not relevant. So when we're talking about products that are locally administered and that therefore are not going to reach systemic exposure, then we do not have immunogenicity information that can be derived as part of the PK study. So here we may need more discussion regarding physical, chemical, structural, functional characterization, or we may even need additional studies if this is not sufficient, which could be phase 3 studies. But as I said again, this really depends a lot on, this is really case by case. It really depends a lot on how you characterize the molecule, on the sufficient amount of studies that you've done to adequately characterize it. This is the one that will guide the need for further clinical evidence, which does not always mean phase 3 studies. It can mean other types of clinical evidence. But yeah, I would say this. And I don't know if anyone wants to add anything.
No, Stefan, I just see there's one question, and I think that may also be an interesting one. Is there parallel scientific advice accepted by EMA for biosimilars? And I'm not quite sure the question goes if it's parallel scientific advice with FDA and EMA, or more products in parallel from the same company to EMA. So maybe we need to
I think they were referring to parallel scientific advice with FDA and EMA. And I think there was already someone very diligently already answered this question.
Yes, and I would say I want to answer that quickly as well in talking. I think yes, it's acceptable because of the guidance reference from EMA and the position paper reference from EMA. But at the moment we're in such a procedure, and we submitted briefing books in parallel to FDA and EMA, and that's absolutely very well accepted because and also preferred from my point of view, by the way, because that gives you the chance to harmonize your applications between FDA and EMA and ensure you have ideally one submission serving all markets, and then have no difficulties and differences in your commercial manufacturing process that you need to adhere to different things requested from EMA and FDA. So please try to align. That would be my recommendation.
Great. There was one more question. What is the regulatory expectation with respect to the characterization of assay cell lines?
That's a good question, because unfortunately, like, real regulatory guidance is only out there for production cell lines, and this is, of course, for the assay cell line, we would not go with the same characterization profile as for the production cell line. But generally speaking, what we did and what authorities always, what was always enough for the authorities is, on the one hand, it's identity. Really, you have to confirm that the cells you're working with are actually the cells you're working with. And this may sound a little stupid, but there is this number out there that I think like between 40 or 50% of all cell cultures are actually HeLa cells and not what we think they are. So it makes sense to actually make an identity. So this would usually be a PCR of your cell line, that you actually have the cells that you think you have. And then, of course, it's sterility. And it's not just microbial sterility, but also test if your cell banks are free of mycoplasma, which is a very important thing. Mycoplasma is like almost like it's not visible. It's very hard to detect, but it can really screw up your assays and really have a big influence on your assays. So this is one important point. And then what we always did, and this just makes sense, is a growth curve and a passage range. So with this set of tests, we usually enough to confirm to the authorities that you have characterized your assay cell line. And also important, do this not just for your master cell bank, do this for all your working cell banks as well. So that's what I would say is covers the regulatory expectations. Unfortunately, again, as I said, at least not to my knowledge, there is no actual guideline out there that tells you what you absolutely must do for assay cell lines. It's just for production cell lines.
And Klemens, would you connect that to the validation package of your bioassay?
Definitely. So what we usually did is our cell line characterization was always in parallel with our assay development and assay validation. So as soon as we had this first vial of our cells in culture, we already started with the development because it just makes sense. And then our cell banks were prepared in parallel to the assay validation. Yeah. The cell characterization of this. Because if you're thinking about passage time, so if you're thinking about this window of how many splittings can I do until my assay does no longer produce a desired result, this takes some time because you will need to split your cells for a certain time until you reach these passage numbers. So start with it early on, because this characterization will be parallel to your assay validation. Yeah. Thanks for this question, Martin.
Elena, I have also in the meanwhile maybe one question for you. You mentioned in your presentation that there is like the specific guidance from MHRA on tailored clinical approach. There is a position paper from EMA and there's a position paper from FDA. Can you explain from your point of view why only MHRA put a definite guideline out there to go with tailored clinical approach, and EMA just went with the position paper so far?
Well, I think it mostly has to do with the fact that MHRA has, one country to kind of harmonize, and EMA has 27 countries to harmonize. I do think, as I said, so I do think that in general for streamlining biosimilar development, everyone is on the same page and everyone wants the same thing. But I do think that it takes more in different scenarios than others. And when we're talking about the EMA, there's a lot still of reluctancy, especially on the clinical side of, default clinical efficacy studies. And there's a lot of residual uncertainties, and the residual uncertainties are coming from much more people than for the MHRA. So I do think that the global thinking is the same. It's just that it's much more difficult to harmonize when you're coming to talk about the EMA than when you're just the MHRA. And I do have the feeling that when the EMA will publish their definite guideline after, the concept paper and after the consultation and everything, I do think it will all be in line. And everyone talks about harmonization of different regulatory agencies and everything. And of course, that would be wonderful, but there's much more things that have to be taken into consideration than just scientific evidence and experience and everything. It's, how each regulatory agency works. There's a lot of things regarding market, and that always makes the process much more difficult. But I don't think it has to do with anything that, I do think that everyone believes in this change.
Yeah, thanks.
Great. I think if there is no further questions, I would like to thank you all again for your participation today. I think it was a very lively and insightful discussion. Thank you so much for joining us. And as it's so close to Christmas, I wish you happy holidays already well ahead. If there's any questions at all, please feel free to reach out to us here at the contact details, or we're happy to share the contact details also of the speakers, of course. And yeah, we look forward to staying in touch. Hopefully we'll conduct a few more webinars next year, and we'll stay very close to this evolving landscape. Thank you very much, and have a great rest of your day.
Thank you
Very much,
Thank
everyone.
you. Bye.
Bye. Thank you.